My name is Professor Oliver Fien. I'm director of the West Coast Metabolomics Center, and we have a monthly webinar series, sometimes with in-person people, depending on if people can travel and whatnot. Not all the time, but sometimes we have in-person or hybrid, actually. And this month, I'm very happy to see Professor Lauren Ashley Cowart. I think you go mostly by Ashley, right?
Thanks.
Okay. She is in the Department of Cellular, Molecular, and Genetic Medicine at Virginia Commonwealth University. Her laboratory investigates mechanisms linking obesity to its downstream pathologies, including adipose tissue dysfunction, non-alcoholic fatty acid liver disease, diabetic cardiomyopathy, and in principle, how obesity or obesogenic conditions, for example, diet, impact the metabolism of lipid signaling molecules, including sphingosine 1-phosphate and ceramides. Yay! That's what her major purpose is. Professor Cowart has, of course, also a history. She used to be a postdoctoral fellow at the Medical University of South Carolina in Charleston. And then before, she did her graduate studies in Vanderbilt University in Nashville, Tennessee, on cytochrome P450s, already in lipid bioactivation of arachidonic acid, and did an undergrad before in Greenville, South Carolina. So she was assistant professor in South Carolina 2005 to 2009, then moved up to the next position, still at the Medical University of South Carolina, got an associate professorship in South Carolina, and then went to Virginia Commonwealth since 2017, where she is a full professor, and I understand also has now other roles, including as a chair of the department, right? Yes.
Yes.
Which is both a burden and an honor. Yes.
It's been exciting lately.
Yes, it's something I defended myself for a long time, to not have too many admin roles if I can avoid it. Okay, so these were the introductions of Professor Cowart, so that we kind of know her a little bit. So again, if you just joined the room, thank you for coming today. And yeah, why don't you share your screen, Ashley, and then go forward.
Okay. Thank you so much for inviting me. I spend my whole life thinking about small molecules, but the hydrophobic ones. So I think a lot about lipids, and it's really nice to be able to talk to many of you who may focus more on polar metabolites. We're actually finding interrelationships between lipids and polar metabolites in terms of sphingolipids regulating components of cell metabolism, and a lot of crosstalk there. So I've also noticed in interacting more with traditional metabolomics folks that some of the lingo we use is a little different. And so it's good to be exposed to your group. So thank you so much for inviting me. I'm going to talk about sphingolipids today, and we're proposing a certain group of sphingolipids as metabolic modulators in heart and liver. So I think you all know about lipids, but the way I think of them is, let me minimize the pictures. I think of them across these classes. We have the glycerophospholipids. Of course, we have glycerolipids, which are diacylglycerol and energy storage in glycerolipids. And then we have sphingolipids, which I think of as being very similar to diacylglycerols, except rather than glycerol, having a serine for a backbone. And I'll show that in more structure later. And then we can't forget the sterols. So together, all of these can be components of different cell lipids. You probably know that different cell types and even different organelles and compartments within the same cell can have very different lipid composition. So it's not just this diagram of a lipid that looks like little more complexity than a plastic bag holding the contents of the cell inside, but these are very dynamic and complicated organelles in and of themselves, the membranes being. So sphingolipids comprise about 10% of total cell membrane lipids. They are required for eukaryotic life. If you knock out the first enzyme in the pathway, it's embryonic lethal. And they have a lot of very diverse functions. So for example, shigatoxin and cholera are receptors for those toxins are actually glycosphingolipids. And immune cell markers are CD17, for example, is a lactosyl ceramide. And then we also have signaling functions. Sphingosine 1-phosphate is a ligand for G protein coupled receptors with really important roles, even clinically, in regulating the immune system. And ceramide is a little trickier. We know that when we modulate it, lots of things happen, but very few protein targets have been demonstrated. And I personally, my personal philosophy is that I think a lot of what we've been calling ceramide signaling may actually be roles of sphingolipids in membranes around membrane-bound proteins and modulating enzyme activities or receptor activities that way. And of course, that's very difficult to study. But we're kind of starting to, I'll show you some data that'll explain why we're starting to think that a little more. So sphingolipid biosynthesis starts out with this enzyme serine palmitoyl transferase, a very straightforward name. Transfers palmitoyl, or the palmitoyl moiety from palmitoyl-CoA onto serine. It generates the first sphingolipid, which is a sphingoid base. It's a one fatty chain. It's the palmitate, 16 carbons, and then the 2 of the serine that are in line. That makes an 18 carbon sphingoid base, and that's by far the most abundant. Then the nitrogen group on the sphingoid base can be acylated to form a 2-carbon chain. Sorry about that. 2-carbon chain molecule that people who are familiar with lipids but don't think about sphingolipids, I say it's a little bit analogous to diacylglycerol, like I just said, but with a serine linking the two together rather than a glycerol lipid. And then from ceramides, this is kind of a central hub in the pathway. Ceramide will serve as the substrate for sphingomyelin or for a tremendously complex number of glycosylated sphingolipids. And this is probably more than 1,000 known classes of compounds in and of itself. So these would be all of the gangliosides, cerebrosides that are really important in the nervous system. So many disease, I am sorry, I don't know what's happening. Many disease processes involve changes in sphingolipid signaling and changes in sphingolipid metabolic enzymes, fluxes, and in the cell kind of landscape and profile of sphingolipids. So what's kind of interesting about this pathway is there's only one way in and one way out. So you come in through sphingosine, serine palmitoyl transferase, but you have to make sphingosine 1-phosphate, which is then acted upon by a lyase that will release phosphoethanolamine and hexadecanal, so 2 non-sphingolipid products. So that is the only way out of the pathway. So this part of the pathway has been of intense interest as well. The rest of the time, changes in the pathway involve shifting things around from one pool to another or remodeling some, changing head groups, changing acyl chains, that sort of thing. So some of the impact of disease-associated processes that how they affect sphingolipid pathways. So we know that beta adrenergic agonists will decrease ceramide production. We also know that many pathological conditions, including high-fat diet, obesity, inflammation, treatment of cells with saturated fatty acids or high saturated fat diet in mice will increase these pools of ceramides, as will less direct, not necessarily a substrate supply mechanism. So oxidative stress, microbiome changes. We also know that hypoxia can result in S1P production. And bile acids crosstalk with sphingolipids as well. And we know that, for example, adiponectin, which is a key hormone for regulating total body energy homeostasis and adiposity, the receptor for that actually has a ceramidase enzymatic activity that can reduce ceramide. So, as I said, the field is a little mechanistically weak on how ceramides do their thing, but we have well-defined roles for S1P, and there are some pretty well-established signaling mechanisms. But I'm sorry, this seems a little out of control. I haven't used exactly the system before. So all of these processes can impact things like skeletal muscle insulin resistance was probably the first observation that if high-fat diet would induce insulin resistance in skeletal muscle cells, but if you blocked ceramide production, that you could attenuate that effect. Similar things have been shown in liver. We know that adiposity in brown adipose tissue, thermogenic adipose tissue can be its activities can be modulated by changes in sphingolipids. And then we know that in a lipotoxic context, for example, obesity, pancreatic beta cells can actually be killed by excess ceramide production, potentially contributing to the progression of type 2 diabetes. And a lot of what I will talk about at the beginning part of the talk will be our work in heart, and different modes of heart failure and how different sphingolipid pathways contribute to that. And then also the second part will be more in liver. One thing also to keep in mind, and this will become, there will be a tie-in later in the talk, is that sphingolipids, like all lipids, are highly compartmentalized. They probably get from one compartment to another through a variety of mechanisms, including vesicular transport, and all of those kind of lipidy intracellular transport pathways. But also there are ceramide transfer proteins, but they're throughout all of the organelles, but in different concentrations in different ones of them. So ceramide synthesized in the ER, but then it's transferred to the cis Golgi or even the trans Golgi, where it takes on head group modifications and goes to the plasma membrane. Catabolism at the plasma membrane generates sphingosine 1-phosphate, which can autocrine signal onto S1P receptors at the cell surface. And then also there's turnover involving the lysosome, whereby pieces of membrane are taken into the lysosome, lipids and everything else is catabolized and then can go back through these pathways. So different cell organelles and membranes have different characteristics when it comes to their sphingolipid profile. So we were trying to look at skeletal muscle insulin resistance in a mouse model. And so we put mice on a high fat diet. We used 2 different high fat diets that I'll describe later. They had different fat sources and different fatty acyl chain compositions. And what we found as we dissected the mice is that the hearts looked a fair amount larger. And that actually ended up being the case. I think I have the more physiologic data on the next slide. But we used a diet based on milk fat, which is very high in C14O and C16O saturated fats. One reason we did this is we were not getting great results with this lard-based high-fat diet. And then I went and looked at the composition of the acyl composition of pork fat, and it's actually quite high in mono and polyunsaturated fats. And there's a body of literature on oleate, for example, 18-1, suppressing effects of palmitate. So I think we hypothesized that by feeding the lard diet, we weren't getting insulin resistance due to that. But what we did find is that when we measured total ceramides, and these are ceramides immediate precursor dihydroceramide, they usually look pretty similar. When we fed the lard diet, we got a lot more total ceramide compared to the milk diet, which pretty much showed no changes. But we only saw heart pathology on the milk diet. So I went back and forth with my student for 3 or 4 months, insisting that she had switched the tubes or mixed up the diets in the animal room. Finally, she convinced me. We were both frustrated with each other. We made up. We're great friends today. But she had been right. There was no problem with anything she had done. The lard diet, though it drove ceramides more than the milk diet was more toxic. So then we decided to look into, well, maybe the milk diet is not increasing total ceramides, but maybe it's producing particularly toxic ceramide species. So we looked for any sphingolipid that increased in the milk diet where we had pathology, but didn't increase in the lard diet where the mice were okay. And what we found is this C14 ceramide. So after we synthesize the sphingoid base, the nitrogen of the serine is acylated, and this occurs through a family of 6 ceramide synthase enzymes, and they all have partially distinct N-acyl chain selectivity. So we thought this is a clue, this C14 ceramide is a clue that this diet may be changing metabolism through one of these enzymes that generates C14 ceramide. And fortunately for us, we did not find robust ceramide synthase 6 expression in our mouse hearts. And so we focused on ceramide synthase 5. So here we have a model of this C14 ceramide. So just to recap, we have our 18 carbon base here, and that's from serine palmitoyltransferase. And then it's been N-acylated with a myristoyl-CoA, the myristate group from myristoyl-CoA to make a C14 ceramide. So it took us a long time to get to this step because of a move and a lot of other things, but we wanted to make a cardiomyocyte-specific null animal in the ceramide synthase 5 because we hypothesized that this mouse could potentially be protected from the high-fat diet-induced diabetic cardiomyopathy, which was changes in structure and function consistent with what's observed in diabetic humans. So we made a tamoxifen inducible, placed them on, so we deleted the gene and then placed them on high-fat diet and did a variety of physiologic and cardiac measures. And we found indeed that our hypothesis was consistent with the role for ceramide synthase 5 specifically in this process. You can see that the controls, male on high-fat diet, we have in the control a very obvious to the naked eye hypertrophy, which is attenuated. This heart looks about like the control diet hearts in the knockout. And we saw the effect in females and it wasn't as robust, and that's consistent with how these high-fat diet models tend to work. The phenotypes tend to be a little more intense in the males, so we weren't surprised to see that at all. And this, not only this difference in size, but also difference in measures of the ventricular volume and the wall thickness, all of these things that cardiologists tend to look at. But the most important thing for this talk is that now all of a sudden I've lost my ability to advance. The most important thing for this talk is that knocking out ceramide synthase. So the ceramide profile gave us a hint it could be CERS5, and knocking out CERS5 protected from the effects. And we can see here, these are cardiac measures where going down is expected on a high-fat diet, but the knockouts are protected. Here, going up is expected. Knockouts are, in this case, not super protected. But it was pretty robust in terms of message for profibrotic programs, including alpha smooth muscle actin, COL1A1, TGF beta. You can see that the high-fat diet in the control mice leads to a pretty strong induction of these, and that's prevented in the knockout. So that's about where we are with that part of the project, but we did look at how energy metabolism might be changed in the heart, because the heart always has to keep pumping. So it's very metabolically flexible. It can use glucose, can use fatty acids, it can use ketones. But we wanted to see how these high-fat diet-fed mice were behaving, the control and then in the knockouts. So high-fat diet signals that energy is abundant, and so catabolic processes will be activated rather than anabolic processes. And we can see here the phospho-AMP-K, which is driving catabolic pathways, is silenced here in the high-fat diet animal, and that's completely prevented in the SIRS-5 knockout on the high-fat diet. So these animals are kind of behaving as if they're in a low energy state consistently. So really, the fundamental way these animals are configuring their energy producing pathways is opposite. So we also found that depletion of ceramide synthase 5 decreased. These are the long chain ceramides that I've already shown you in the previous paper. I should have shown this slide first, I'm sorry. But we do see that the high-fat diet is driving C14 and C16 ceramide, and that knocking it out does ameliorate. And these residual ceramides are probably, not complete penetrance of depletion of the gene. We also know that a lot of different cell types there are a lot of different cell types in the heart. So cardiomyocytes make up only about 35% of cells by number in the heart. So now I want to shift away from ceramide per se and talk about the generation of the sphingosine base. That's the first step. So this is what I showed in one of the first slides. Let's talk a little more about serine palmitoyltransferase. It is a multi-subunit enzyme, and it occurs in a dimer of tetramers. And the catalytic subunit is SPTLC2 here. SPTLC1 is kind of a scaffold for the dimer. And then we have some smaller regulatory subunits. ORMDL is an interesting one because it senses membrane ceramide and then will provide negative feedback to shut down SPT, this enzyme. So it's a nice feedback mechanism that's reminiscent of how SREBP regulates cholesterol biosynthesis in response to low membrane cholesterol. So it's very similar paradigmatically. And then these small subunits are optional, but they can have some impact on in acyl chain selectivity of the complex. But by far, the most abundant and the classic substrates are palmitoyl-CoA and serine, making that 18-carbon sphingosine base. I will start to refer to things as D18, and others, and the D there is for this dihydroxy. So this is a classical dihydrosphingosine. So then in acylation with the CoA through the action of ceramide synthases generates a ceramide. And this classical ceramide can then go and be distributed among all downstream sphingolipids. So a new subunit was discovered, I got 2006 or so. The reference is probably, oh yeah, there, I just can't see that on the screen. And it's called SPTLC3. And what Torsten Horneman, who discovered this and is a friend and collaborator, he noted that if SPTLC3, it could substitute out for SPTLC2, and then it really relaxed the selectivity of the enzyme. So not only could this enzyme of the 1,3 configuration use palmitoyl-CoA, it could also use myristoyl-CoA. And a lot of our early work focused on that. And then more recently, it seems that it could potentially use branched chain fatty acids, very long chain fatty acids. And I think the work here is just beginning to get its surface. We're just starting to scratch the surface. So if we use different CoAs to condense with serine to make our sphingosine base, we get different sphingosine bases. So if we use myristoyl-CoA, we have 14 carbons from myristate and 2 carbons from serine, making a 16-carbon sphingosine base. Similarly, 17. So if we use a 15-carbon fatty acid and the serine, then we will make a 17-carbon sphingosine base. So there is kind of a dogma because fatty acids are synthesized in units of 2 and mostly degraded in units of 2, that all fatty acids are even chained. And we've had critique from reviewers, where are these odd chain fatty acids coming from? But people have begun to more get on board with the idea that these things are appreciably there in humans once we started doing untargeted lipidomics, and the untargeted lipidomics would then show things we weren't necessarily looking for, including odd chain acyl chains as components of lipids. So where do these come from? Well, peroxisomes do some alpha oxidation, which would remove a single carbon. But one thing that's getting a lot more attention now is Odd chain, short chain fatty acids from the gut microbiome can be elongated through fatty acid synthase. So we could be elongating odd chain starters in groups of two. The other thing that I'm very interested in is this 17 carbon dihydrosphingosine a 16 carbon methyl branched? And that is also a possibility. And we've worked out some techniques to distinguish those by really lengthening our liquid chromatography and separating a methyl branched from a linear. So all of these sphingoid bases, at least the ones that we focused on mostly, but I can tell you 16, 17, and 19, some others I haven't shown, they can be distributed throughout the pathway in all of those different downstream sphingolipids very similarly, but the rates of flux are different. And so how different bases go into different pathways at which rates is something we've become increasingly interested in, and we're just starting to look at that. But what's interesting to us about SPTLC3 is that GWAS studies have linked SNPs at SPTLC3 to all of the diseases that we know sphingolipids are involved in. So cardiovascular, liver, psychiatric, and neurodegenerative. So we know sphingolipids are really critical in the nervous system. And then many of us have been studying liver and cardiovascular disease with a focus on roles of sphingolipids there. So this led some strength to the notion that this could be potentially important in humans. And so when we looked in our mouse hearts, we actually found that there was a high degree of SPTLC3 expression, specifically in the mouse hearts and not in a very similar tissue, skeletal muscle. So we began to wonder if SPTLC3 could be playing some particular role in the heart, in the myocardium. So since then, our studies have demonstrated across the board, and I'll get into more details on these projects as I go on, that SPTLC3 tends to have pretty low basal expression, but we found numerous disease processes induce SPTLC3, including cardiac ischemia, diabetic cardiomyopathy, alcoholic and non-alcoholic liver disease. Thermogenic adipose also expresses SPTLC3. So even though it's not very broadly expressed, it is inducible, and we think it's playing particular roles in cells under these conditions. So to get back to the mouse model of our heart disease, we thought, well, this would be a lot stronger if we saw SPTLC3 in human. We had been using diabetic models in mice, so we asked our collaborator, Fadi Saloom, who had the human samples, we asked him for heart failure patients with ischemic cardiomyopathy is what he had. And we said, well, can you look back at the records and give us half diabetic, half non-diabetic? And what we found was that the diabetes was not necessarily regulating SPTLC3 more. It was just the fact that they had ischemic cardiomyopathy. So we wanted to see then if the sphingolipids in those hearts contained the atypical sphingoid bases. And we did find that, for example, the dihydrosphingosine sphingoid base of 16 carbons was increased, as well as the phosphorylated derivative increased in the ischemic patients. And actually, the 18s were slightly decreased, the 18s of these particular species. And this is more or less observable throughout the pathway. So we set up this same model of this disease in mice, which is kind of occluding an artery that provides oxygen to the heart. Lad ligation is what we call it. And we found that in contrast to humans, the mice do express basally in the heart, which is consistent with what we had found before, but that this was induced in lad ligation. But what was very exciting to us as well is that the pattern of the change in sphingolipids was identical in the mouse model as it was in the human disease. So we felt very confident moving forward studying this mouse model and indeed SPTLC3 in this project. So we made we had to actually flox this allele. It took us a long time. It was tricky, but we developed our floxed allele and then were able to make a cardiomyocyte-specific knockout mouse. And we found, to keep a long story short, that these mice did gain protection from that lad ligation, both in terms of the observable fibrosis, the dilation of the left ventricle, which is not as severe here, other measures of function, including fractional shortening. But most importantly, I guess the survival rate was much better in the SPTLC3 null mice compared to controls. So, of course, we had no idea mechanistically how in the world this could be happening. So we did an RNA-seq, just a bulk RNA-seq, and we found that many of our top pathways involved metabolism, and particularly mitochondrial metabolism, or things associated with mitochondrial metabolism. So we took a look at these with the collaborator, Sushil Mahata, by electron microscopy. And it's pretty obvious at the 40,000x magnification. These cristae are organized so beautifully. This was just kind of a profound thing. I don't look at cells a lot. I think more about grinding up things and measuring stuff. But I just think this is so beautiful, the way that nature has developed. On the other hand, if you look in the knockout mouse, you see much fewer cristae, and you see they're not very well organized. And that was consistent with loss of mitochondrial function, including loss of polarization in the knockout. So we're having a fundamental mitochondrial defect. So these mice haven't been challenged with high-fat diet or lab ligation. These are just basal. So what we found looking at the electron transport in the mice, because it's very critical in the heart to be able to have a consistent supply of ATP, is that in the wild type, we had a lot of complex 1 activity, but we reduced the complex 1 activity significantly in the absence of SPTLC3. Complex 2 was not as affected. These are just two populations of heart mitochondria. It's not important for this talk. Complex 2 and complex 4 were not impacted to the same extent as complex 1. So complex 1 and 2 feed electrons through ubiquinone coenzyme Q to complex 3 to fuel the ATP, to fuel ATP synthesis. But the actual enzymatic function of complex 1 is to take the electron from NADH and transfer it to the ubiquinone, and that generates NAD plus that has its own impact on mitochondrial sirtuin. So even though we're talking about ATP production, there's a lot of modification of metabolism just when electron transport chain is modified. So we can see here that we measured the in vitro activity of this enzyme, and we found complex 1 just wasn't performing in vitro. So we have a fundamental complex 1 defect in the mitochondria there. And so we thought, well, this is strange. How could this be happening? We don't know of any mitochondrial expressed ceramide signaling targets. Let's just pull down complex 1 light detergent and see what membrane lipids come down with it. And when we did that and followed that by targeted mass spectrometry, we found that lactosyl ceramides with the SPTLC3 derived bases were very abundant in the wild type, but not in the knockout. And in contrast, these would be the standard everyday sphingolipids, so not necessarily SPTLC3 dependent. They didn't show the same effect. So these lactosyl ceramides are driven by addition of a glucose head group to ceramide, followed by addition of a galactose head group to ceramide, and we result in a lactosyl ceramide. And it's accomplished by sequential action of these two enzymes, UGCG and B4-YALT5. And what we're really excited to see is that expression of both of those enzymes were highly expressed in the human left ventricle tissue in the heart failure patients. So we're really thinking this could be relevant to human health. And then we're left with the question of SPTLC3 was almost undetectable in normal human heart, but there was appreciable expression in normal mouse heart. And then it dawned on us that mice have a very high rate of ATP turnover because their heart beats so fast. So we think that might be why SPTLC3 is there to boost the activity of complex 1. So, as I'm sure you all appreciate as metabolism specialists, that different organs have different metabolism. So, a healthy heart has a high continuous ATP demand. It's fatty acid oxidation dominant in a healthy state. It can use ketones when fasting, and it does not store much glycogen. So, in contrast, a healthy liver serves as a central metabolic hub, kind of, I think of it as gobbling up any metabolic intermediate and trying to feed it into the TCA cycle to make acetyl-CoA, to make glucose, to make fat and package. It also stores glycogen, regulates glucose, and it also performs gluconeogenesis, unlike most other tissues. And rather than using fatty acids for energy, it's much more likely to be synthesizing synthesizing fatty acids. So we thought mitochondria have very different roles in liver. Would depletion of SPTLC3 in mouse liver cause a similar outcome? So we made the liver-specific SPTLC3 knockout. We did find some basal SPTLC3 expression in liver of mice as well, and our knockout was successful. And we took a very similar approach, first of all, doing just a bulk RNA-seq. And again, we found many of these pathways that are pointing us to mitochondria, not only in electron transport chain, but also structures and all of this. So I think these categories arise in part because the pathways are overlapping of what's driving what, but it all pointed to a mitochondrial defect. And that was shown, replicated as we had shown in heart with a decrease in mitochondrial function. So it was pretty similar to heart up to that point. When we looked at mitochondria, the ultrastructural defect was it was much stronger in that we could barely see Christie in many of these mitochondria. We also saw that basal glycolysis was extremely high. So a liver's job is to make glucose for the rest of the body, not to consume it. So this is very atypical for liver. And then we also saw that mitochondrial oxidative metabolism was decreased. And the mitochondrial area and Christie density were also modified. So we definitely have some evidence here for mitochondrial damage. However, what was very interesting to us here is, in contrast to heart, complex 1 enzymatic activity in an isolated complex 1 from these livers was not inherently impaired. So we do have decreased ATP, decreased NADH to NAD ratios, but if we actually look at the complex 1 activity, it's really increased in the SPTLC3 mouse. But what we do see is a decrease in the transfer of complex 1 electrons from complex 1 to complex 3. We aren't sure of the mechanism for that, but we did then measure CoQ9, which is the electron carrier in mice. In humans, it's CoQ10. But we saw a small decrease, maybe generously a 20% decrease in CoQ9. We are not sure if this could account for the decrease in ATP production and the mitochondrial phenotype that we find. We're still looking into that. But depleting SPTLC3 dependent lipids really impacts the mitochondrial metabolism in liver, just as it did in heart. However, when we pulled down complex 1 in liver, we didn't really find major differences around complex 1 per se. But what we did find is that when we isolated mitochondria, we found very large differences in the lipidome. So here we have liver homogenates and the mitochondrial fraction, and here are just the markers showing the degree of purity we were able to achieve. When we did untargeted lipidomics of the liver homogenates, we got pretty much no separation of the wild type versus the knockout. And you can see that in the way the clustering isn't really tightly aligned here. And the PCA plot shows no big difference. On the other hand, once we looked at the mitochondrial fraction that we had isolated, very clean separation of groups. And we can see that the knockouts, they're not only missing SPTLC3 dependent sphingolipids, which aren't depicted as such here, lipid classes are grouped together here, but a lot of other lipid classes are impacted. So we think that the mitochondrial defect in liver is more a result of shifts in the whole metabolic landscape of the mitochondria itself. And so we wanted to look at what sphingolipids were impacted. And if you look at the sum of the ceramides in the whole liver homogenate, we really don't see a change in these that would be SPTLC3 dependent. So a 16 carbon sphingoside base, 17 carbon, 19 and 20. The 18 carbon is the standard one that would come through the original, the 1,2 complex, not through SPTLC3. However, if you look at the mitochondrial fraction, that's where you see your SPTLC3 dependent lipids decreased. So we think somehow those lipids in the mitochondria themselves are playing some role in regulating the lipid content of the mitochondrial membranes themselves. So we thought maybe these mice, could they be protected in the high-fat diet situation? And we just kind of, if what you have is a hammer, everything you see is a nail. We had done a lot of high-fat studies. We didn't have the strongest rationale for this, but we decided to do it anyway alongside some other things we were doing. And we did find an interesting outcome. So we found that hepatocyte, we found that the animals were protected from high-fat diet induced elevation in fasting glucose. But through a long series of experiments with yielding negative results, we found that that was not due at all to a protection from non-alcoholic fatty liver disease or insulin signaling. It was that glucagon, gluconeogenesis, which is stimulated through glucagon, was impaired. So when glucagon is released from the pancreas, that is a signal for the hepatocyte to make glucose for the rest of the body, and it binds its cell surface receptor on the hepatocyte that activates adenylyl cyclase, which is membrane associated, leading to production of cyclic AMP, which then activates protein kinase A and all of the downstream pathways required to stimulate gluconeogenesis. And through a series of experiments, we found that in a wild type glucose treated hepatocyte, we got robust cyclic AMP production in response to glucagon, and that was attenuated about halfway by removal of SPTLC3. And consistent with that, we got downstream signaling that was consistent with attenuation of glucagon signaling, including PCK1 dephosphorylation and even message regulation of these critical enzymes in gluconeogenesis, and even GLUT2, which exports glucose out of the hepatocyte into the circulation. And we knew that this was occurring all at the receptor and adenylyl cyclase end of things, because if we treated with a soluble cyclic AMP analog, everything looked similar in wild type and mutant. So we knew that up here somewhere is where we were having our SPTLC3 dependent defect. So sphingomyelin is very abundant in the plasma membrane. And so we first turned our sights to sphingomyelin. Could sphingomyelin be different? Could there be some SPTLC3 derived sphingomyelin that is important for glucagon signaling? And sphingomyelin presents a little bit of a challenge in targeted lipidomics because the 16 carbon base, 18 carbon acyl chain sphingomyelin is isobaric to having the 18 in the other chain and the 16 in the other chain. So these are both the same. There is a pseudo MS3 strategy that's been published, but it's a little tricky. And in the meantime, all of this, we were coming up on this, our friends at Stony Brook developed a really nice method of looking specifically at plasma, outer leaflet plasma membrane sphingomyelin. So what they established and validated is that you could treat your cells how you wanted. You could fix your cells similar to how you might for imaging. And then you can treat with bacterial, purified, commercially available bacteria sphingomyelinase, and that will release the ceramide moiety from the sphingomyelin. Then you can measure the ceramide, which does distinguish between, which does fragment to show you what's in the base and what's in the N-acyl chain in the MS2. So you've kind of answered two questions. You can assume the ceramide that's produced comes from sphingomyelin because it only came after you treated with sphingomyelinase, and then you get the N-acyl chain composition and the sphingoid base composition. And what we did find is that in the outer leaflet of the plasma membrane, when assessed through this method, we did see decreases in the SPTLC3 dependent sphingolipids. So we're not sure of the mechanism, but we do know from there are a couple of really nice JBC papers back in the 80s demonstrated that adenylyl cyclase activity is very sensitive to its membrane environment. So we're thinking, this result coupled with the lactosyl ceramide and the complex 1 story in the heart we think that a lot of what we may have been calling sphingolipid signaling may be distinct pools of sphingolipids around and interacting with membrane proteins and impacting their function. And that is an idea that it's a little bit controversial because I think everyone wanted ceramide signaling to be, ceramide molecule protein target downstream signaling, kind of like the diacylglycerol protein kinase C story. But I don't think when we get to these mechanisms, they're all going to be that paradigm. And another little piece of unpublished data I wanted to show you is that, we looked at the plasma membrane enriched for the SPTLC3 dependent sphingomyelins. We showed the liver having the SPTLC3 dependent ceramides in the mitochondria. We also have a friend down the hall who pulls down lysosomes. So we thought, let's see if we have if we have a concentration of these atypical sphingolipids in the lysosome. So this is the whole cell distribution of sphingoid bases in ceramide. So you can see in the whole cell, 96% of the ceramides have your standard everyday 18 carbon base, 2% atypical. However, if you pull down just the lysosomes, we see that we've got much more D16-1 and D17-1 in the lysosome. So we don't know if those are there because they're degradation products of more complicated lipids or if they play a specific role in lysosomes. We just don't know. But now we have several lines of evidence that different organelles have different concentrations of these atypical sphingolipids. And even though they're not very abundant, we think they're important for what they need to be doing and where and when they need to be doing them. So following up more on the liver story, we noted that we had mouse models, our high-fat diet-fed mice, and also alcohol-fed mice, both had strong SPTLC3 expression in the liver. So we wanted to look at liver cancer in humans because both of these conditions, fatty liver disease, either alcoholic or non-alcohol related, are both linked to an increased risk of hepatocellular carcinoma. So that was demonstrated, supported in part here. We looked at a normal hepatocyte cell line and then 2 liver cancer cell lines, which had a much more robust expression of SPTLC3. We got some samples from our tissue bank here, looking at human liver tumors versus adjacent non-tumor tissue. And we find that in about half of them, we get a pretty robust increase in SPTLC3. And then there's some online database information that high expression of SPTLC3 is linked to low survival. And I believe this is a female specific phenotype, but anyway, it doesn't, all of this goes to support that SPTLC3 may play some role in cancer. And we also found that when we knocked down SPTLC3 in these liver cell lines, they grew a little slower. So that led us to think about, could we develop small molecules that would target the SPTLC1-3 complex, but not touch the SPTLC1-2 complex? So our collaborator in a drug development group at University of Louisville worked with us on modeling the 1-3 structure on the 1-2 structure that's known. So there are 2 cryo-EM structures of the 1-2 complex in the literature. SPTLC3 and SPTLC2 are, I think, around 80% identical at the amino acid level. So finding differences between them was going to be difficult. Rob first examined the catalytic site, and they were essentially indistinguishable between SPTLC1 and 2. However, he did say, there are these 2 pockets that could work in an allosteric manner that occur in SPTLC3, but do not occur in SPTLC2. So he did an in silico screen of over, I think, 5 million compounds. So in Kentucky, and probably you can do this in California, we aren't there yet in Virginia. In Kentucky, an academic institution can use the whole computational power of the state of Kentucky over the weekend. And so he ran this over the weekend and came up with a list of potential ligands or potential small molecules that would bind. We bought the top 50 from a chemical library in Ukraine, and he tested them for binding, and we selected the top 4 binders. 52 ended up having, we all the binders we looked at MTT assays, so measuring cell death in HC3716s are our normal cell lines, but then HepG2 and Hus7 are the SPTLC3 high expressors that are the cancer cell lines. And we found that the effective concentration for cell death for HepG2s and Hus7s is somewhat lower than for the wild type cells. So we need to be working on this compound and developing more analogs. We're on our third round of derivative structures, and some of our top candidates now have much better therapeutic windows, meaning they're work at much lower concentrations in the cancer than in the control. So we're really excited about that. We have a provisional patent on the first the idea of using that molecule, and then we're working on patenting our family of molecules that we have now with our, developing our Marcouche structure and all of that is part of the process of drug development and patenting. But we wanted to check if treating AR52 would using our inhibitor would decrease the SPTLC3 dependent sphingolipids in vitro, and we do find that at 1 micromolar, we get a third to a half decrease in these atypical lipids. There's definitely a trend in decreasing the normal lipids, but SPTLC3 complex can also use palmitate. So we don't know if it's just less significant but trending because we're depleting a little bit of that activity, or if the compound is actually impacting SPTLC2. The way we are going to address that is going to rely on in vitro assays. So we're getting our expression system up and running now. We did a few more kind of cancer biology classical experiments, a migration assay in Hus7. So you take a confluent layer and make a scratch and then see how long it takes or how well they fill it in, indicating cell division with and without the compound. And we saw here that the AR52 really decreased the density to which these scratches could be filled in, and that's quantified here. And this is also a cell migration assay, and everywhere you see purple are the normal cells. So AR52 is really decreasing the number of cells. So we're continuing to do these kinds of assays, but our fundamental question, one of the fundamental questions is this happening through mitochondrial metabolism? Because in liver and in heart, SPTLC3 modifies or is required for mitochondrial metabolism. So this was interesting because one question we often get is, what does SPT3 do that SPTLC2 doesn't do? Is this really specific? And we do find knocking down SPTLC2 in our liver cancer cells decreases mitochondrial function a little bit. There's a trend there, but knocking down SPTLC3 has a much more robust activity. And we were excited to see that in the Hus7 cancer cells, 52 behaved very similarly to knocking down SPTLC3. And we also found, which is really promising, that in the normal cells that don't express much SPTLC3, the compound doesn't change the shape of this mitostress test curve. So we think that this is pretty promising, and I never thought that I would be involved in drug development when I started this, but when we started getting these results, we realized it was maybe even our charge to investigate whether this could be useful. So as far as the cancer part of the talk goes, this is the model we're working with, that in cancer we have a high SPTLC1 and 3 complexes due to elevated SPTLC3, that we're making 16 and 17 carbon bases that make the atypical sphingolipids. We also, I didn't talk about MYC today, but c-MYC is highly expressed in a lot of liver cancers, and we know that c-MYC does upregulate SPTLC3. And if we can target specifically the SPTLC1-3 complex with this was our initial lead compound, that we can block production of these lipids and change mitochondrial metabolism in a way that would inhibit cancer growth and survival. So in overall conclusion, though the contribution of sphingolipid production to heart function is broadly appreciated, many people have looked at ceramides in the heart. We were the first to really dissect out specific pathways, including the ceramide synthase 5 pathway. Um, just want to drive home the point that the ceramide synthases drive the acylation of the sphingosine base, but serine palmitoyl transferase composition of that multi-subunit enzyme is what's driving the initial product and what the chemical nature of that initial product is. But both of these contribute to cardiac dysfunction, but in distinct ways. Distinct sphingolipid species contribute to changes in cell energy and mitochondrial metabolism through signaling, through direct impact on OXPHOS, electron transport chain. And one thing that was very interesting to us is though SPTLC3 lipids impact ATP production in both heart and liver, the mechanisms seem different in that the heart has a fundamental enzymatic defect and complex 1 seems to associate with lactosyl ceramide. Liver, there is no specific SPTLC3 dependent lipid associated with complex 1, and there's no inherent complex 1 defect in vitro, but there are gross changes in lipid composition of the mitochondrial membranes themselves. And one more take-home message is that SPTLC3 lipids, they haven't been studied by many groups because they are so low abundant, 100-fold less, 1,000-fold less abundant than the most abundant standard canonical sphingolipids. So many people haven't studied them, but I've showed you a few cases here, the lysosomes, the plasma membrane outer leaflet, and the mitochondria, where these specific lipids are more abundant and seem to be concentrated. So we think that these lipids are highly compartmentalized and they serve organelle-specific functions under specific cell challenges. And also, more recently, the drug discovery component, we think SPTLC3 could potentially be targeted to prevent the metabolic reconfiguration that facilitates cancer growth. With that, I will thank Miriam and David, who have done all of the liver work. Anna did a lot of the heart work, former members. And then I also want to, sphingolipids is a small field, and people who work on serine palmitoyl transferase is even a smaller field. I think myself and these 4 other individuals probably are the most obsessed with SPT of anyone in the world. And it's great that we get to see each other at all of the sphingolipid meetings and sit around talking about SPT. And many of our ideas have come up through these conversations. So I just like to acknowledge my colleagues. The sphingolipid field is very, very friendly and very collegial. We've gotten, a lot of good ideas from these people. And then our VCU lipidomics and metabolomics core is growing all the time. We look to your facility as an aspirational model. I really appreciate being invited to speak with you. And then other cores we've used and other collaborators we have. And with that, I'm happy to take any questions.
Thank you so much, Ashley. That was fascinating, very detailed, enlightening. I really liked your enthusiasm for that research. Now, there's a number of questions. Some of them are basic questions, others are a little more detailed. So let's start from the top. In untargeted lipidomics data sets in general, how can we distinguish functionally beneficial versus detrimental ceramide species? And are these differences primarily determined by fatty acyl compositions, or do specific ceramide synthase enzymes play a more critical role?
That is such a good question. I think that it has to do, so first of all, from the untargeted lipidomics, from the lipidomics, you just get a hint about what the enzyme could be. And then you have to go back to your in vitro situation, I think, or cell or animal situation to test the specific isoforms. One thing I didn't emphasize is that ceramide, but I did mention it, ceramide synthases 5 and 6 can both make C14 and C16 ceramide, but numerous studies have shown that they do not mediate the same biology. So I am wondering, many of us wonder if it's cell compartmentation, if it is interacting partners. We just don't know, but I think it has to do in some cases with the lipid, but in some cases it might have more to do with the enzyme. So very excellent, relevant question.
So then Shen continued and she asked, how about other ceramides like T18-0, D18-2? Are these not found in cells and lysosomes or due to targeted assay not looking beyond?
That is so great. So we have a skin project where we see all of those diverse species that you're talking about. The diene, so the D18-2 sphingoid base, The phyto, which is the T, D18, skin has a really complex sphingolipidome, but most of the cell systems we look at, it varies, but we don't often find those niche sphingolipids in cell culture systems in homogenates. I think probably like some of the lipids I showed here, they're where they need to be when they need to be, but they're not always around in every tissue or every experimental system. And you know, these we weren't even really broadly aware of these until people started doing untargeted lipidomics. So the field is still, I mean, it was just 3 years ago. Someone came up to me and said, "Hey, do you see 17 carbon sphingosine bases? What the heck?" So we're at the very beginning of trying to figure out where they come from and the functional significance of them.
Yeah, and in addition of that, there's always the risk of misannotations, of course, which we didn't even talk about.
Yes, yes. You know more about that than I do, but yeah, we do tend to follow up with targeted using internal standards
Yeah,
when we can.
it is interesting to see that ethanol increases SPTL3. It also blocks serine synthesis. Do we know if SPTCL3 can utilize aniline, a glycine alternative substrate for synthesis of the other atypical sphingolipids?
Excellent question. We're so interested in that. We've spent so much time on the CoA that we have not addressed the amino acid, and I'm actually really impressed that that is even on your radar because it's a very small niche literature. Point mutations that are associated with human neurological problems change the and those have been identified in SPTLC2, change the substrate utilization on the amino acid side, leading to these atypical, other kind of atypical lipids, the amino acid atypical rather than the acyl. And we don't know what the flexibility is for SPTLC3. Out of the 5 people that are working on SPT, I am the one who's most interested in SPTLC3. So I'm trying to do things as fast as I can.
Okay. Does the SPTL3 support the non-classical sphingometabolism, including deoxysphingolipids?
Yeah, same question.
Same question. The deoxys would come from alternative amino acid utilization.
Okay.
So
Yeah.
very spot on questions.
Let me just pop in
Okay.
there for a second. So John Newman here, that's who I asked that first question. Me and my grad student are working in a project where we've just put online methods to measure deoxyceramides,
Okay.
and we've been seeing in essentially tracking cardiovascular risk and alanine to serine ratios in open plasma in clinically healthy people
Oh, wow.
are showing shifts in deoxyceramide levels. So it would be really, really interesting to look at the mitos on your mice and just see if there's deoxyceramides showing up.
Yes.
So we can provide you all the mass transitions if it's something you're ready to do, or we could shoot a couple samples if you're interested.
Yeah. Yeah, that would be interesting. Yeah, for sure. That's fascinating.
Okay.
So it was in human serum?
Yeah, plasma. Yep,
Human
open
plasma.
plasma in the circulation, fasting plasma.
I believe we have done untargeted on our mouse plasma lipids. And if so, I will look and see if we find the deoxy.
That'd be great. I'd love to know.
Yeah, Meng
Yeah.
Jiang, just as on camera, do you want to add on to that?
Yeah, I just want to introduce myself. I'm Dr.
Yeah.
John, a PhD student, and
That's
also
my-
ask the same question at the same time about the oxyphingolipids.
Okay, good.
Yeah.
So then we continue. So Shen asked, seemingly SPTL3 expressions is turning on upon series of disorders, while out in normal conditions is the SPTL3 SPTL2. Is transcriptome data supporting the higher protein expression too?
Yeah, we've done proteomics to actually been pulling down 1 and looking at how much 2 versus how much 3 we can co-IP. But yeah, the transcriptomics data supports that. What we're finding is that all of the pieces of data from all of the different systems are starting to coalesce around this idea that SPTLC3 is induced in rapidly proliferating cells. We think actually that the SPTLC3 in the liver disease models is because of damage and potentially repair of the damage. And then the cancers would be that same process. So that's kind of our line of thinking on that, along that right now. But it can be challenging under normal conditions to detect any SPTLC3. However, I showed you a couple of cases where depleting it did lead to a basal phenotype. So we don't know if in those cases the animals have somehow adapted around it, which has led to that phenotype. We're working on making a tamoxifen inducible SPTLC3 knockout in the heart, because we think if your heart from embryo or to mouse pup, if your heart is not using complex 1 effectively, there probably have to be a lot of metabolic adaptations. So, that's the caveat with the system we used. I think hopefully the inducible knockout will let us know more the acute effects of loss of SPTLC3. Then also we are using some viral models to acutely overexpress and see what happens.
The next question is on the on the overall effects that you had alluded to, that it's not just the signaling, but rather the membrane composition. So you alluded that specific ceramides could have an effect on protein function via contact at the membrane. What type of interactions do you think they are with those ceramides? Do you know if some method could elucidate these interactions specifically?
Yeah, so I am recently appointed chair, and one of my first hires is going to be someone who can do molecular dynamic simulations. And in my package, I'm also getting a GPU cluster to facilitate that. So there really are not a lot of great techniques to study this except computational and theoretical right now. Making a membrane in silico and changing the lipid composition around a protein for which the structure is known. And then using that to develop hypotheses about specific amino acid lipid interactions, making mutants and seeing if that follows. I guess the next step gets really rigorous with purification of those enzymes and reconstituting them in different model membrane systems and vesicle systems with different concentrations, including nanodiscs. I'm getting really interested in our structural biologists here use nanodiscs a lot for membrane protein cryo-EM. I think we could probably use them to test we definitely could use them to test how different membrane composition impacted structure. I think we might be able to look at how it impacts catalytic activity as well.
And I have two simple questions. One from John. What was the percent of milk fat in the milk high-fat diet?
It was 60% kcal from basically butter fat, like
Wow.
oil. And then, well, because we're comparing it to the lard, the standard
Yeah.
lard obesogenic diet, which is 60% kcal from lard. So we had to keep it the same. No one eats that much fat. Well, some people,
Right.
I don't know.
Sticks of butter. That's awesome.
Yeah.
Yum.
And then I have another simple question. When you talk about lard to people, they usually make a face.
Yeah.
So
Yeah.
the simple question is lard less bad than its reputation?
Exactly. Yeah. It's very high in oleate and linoleate, which is an essential fatty acid that's important for cardiolipin, as you probably all know. So, yeah, that was a funny outcome. But then, most traditional diets of people have evolved over time to be kind of healthier, like traditional diets. And I think there are places in China and then also in South America where the traditional fat source historically has been lard. I think maybe the problem with lard might be the cholesterol content, but dietary cholesterol is not bad for everyone. It depends on your own cholesterol metabolism. But yeah, we've spent a lot of time thinking about that.
Yeah, it's a funny side thing, right?
Yeah.
So coming back to more serious things, Shen asked, when it comes to mitochondrial dysfunction, when knockout SPTL3 does it have any effects on acylcarnitines and cardiolipins?
Yes, it does. I didn't show the detailed heat maps for the untargeted in liver because y'all know what those look like and you can't really get any meaning out of them. So I'd grouped them together in classes, but I think acylcarnitines were there, but I know some acylcarnitines are impacted. We also did some labeling for the gluconeogenesis, and the TCA cycle has a pretty significant impact as well. There's an impact on TCA cycle. So it's not only ETC, I just focused on that for today's talk. But my point is, in response to your question, there is a broader impact on overall mitochondrial metabolism beyond the electron transport chain, which I think is what you're asking.
So then we have a more a wide-ranging question from Jonathan. Can you speculate on how some of the pathways and mechanisms that you have interrogated may be important for sphingolipid pools, concentrations in extracellular compartments, including peripheral circulation? Has any of that work in this area done before? And do we know what SPLT3 expression looks like in humans with inborn errors of metabolism like Niemann-Pick?
Oh, that would be really interesting. I could speculate. We're looking at you can kind of get some idea on SPTLC3 versus 2 dependent lipids by what they turn into down the line, because like if it goes to the cis Golgi, you're going to make SM. If it goes to the distal Golgi compartment, you'll make glucosylceramides. Because of the way they're distributed throughout the rest of the pathway, we think that there is that they're routed differently through the secretory pathway. And in our mouse plasma, we get the proportion of the D17 that ends up in the mouse plasma is a lot higher. The delta, the change is much bigger than the 18. So that would definitely argue that there is 17 coming out in extracellular vesicles or as components of lipoproteins. And that's actually a sub-aim in a grant that we're trying to develop. So yeah, great question.
Jonathan, you want to, you just lit up your camera. You want to elaborate or have a follow-up?
No, that's great. And then, yeah, I'm a clinician by training. And so I'm always wondering about what the extreme clinical phenotype looks like, i.e. for patients with inborn errors of metabolism, like Niemann-Pick, where sphingolipid metabolism is perturbed, what things might look like. And if your group or any other groups have looked at that, just from like an observational kind of case series perspective.
Yeah, we haven't. And I think that is a very exciting question. We've done with the geneticists here, we've got a good group of human geneticists, and we've done a PheWAS for SPT3 mutants, like, looking for phenotypes that are associated with human mutations. And we're kind of finding strange things, more like embryonic development phenotypes. But there's a lot of psychiatric phenotypes, which falls in line with the importance of sphingolipids in the nervous system. And then also a lot of that's driven by the kinds of patients that you have in your databases. So in terms of the human, I'm trying to think of the most human relevant stuff. But I think that's the heart samples.
So let's continue with the clinical questions here. Rabia asks, do you know anything about whether SPTL3 plays a role in polyneuropathies? And also, I would like to extend that question. In our own studies on pain that we recently completed, we also see that specifically 18-2 ceramides were also involved in, circulating. We're also involved in, pain development. So what do you think about those?
I think that sounds very reasonable. There's been some work done on sphingosine 1-phosphate in pain signaling, but to my knowledge, not as much on the ceramides. So I think that is really exciting. We do have kind of a side project with the collaborator who's interested in nociceptin and pain perception, and in a nociceptin, manipulating that system does have some impact on sphingolipids or vice versa. I can't remember offhand. I know this is not very helpful. I'm just trying to think of anything I might know related to your question, but not many people study SPTLC3. So I'm glad that you guys are so interested. Maybe you can look at these atypical lipids in your own systems and help answer some of these questions. There's just a lot of work to be done.
Yeah, so since because they are all involved in signaling, or maybe not, maybe just in the function of the protein complexes, I mean that's why people get more and more interested in sphingolipids and ceramides, right?
Yeah,
Um,
agree.
and clearly also they're involved in pain. I looked at the literature yesterday and I found quite a number of good papers.
Mhm.
Um, so
Oh, good.
there's
Good.
clearly a story to play there, and I think Rabea was right on topic, right on target. Whether or not it's SPTL3, that's a different story, right?
Mhm.
Of
Well,
course.
now maybe these are on your radar, so when you get your data sets in untargeted, you'll recognize these atypical ones and then maybe help make some of the connections that need to be made. That'd be great.
Yeah. Okay. So then we have a question from Shen again. Since milk is higher in myristoyl fatty acids, does it all increase the myristoyl-CoA since higher C16 ceramides and maybe increase also I guess the importance or so of SBTL3 expressions?
Yeah, I think that was our hypothesis because when we found the C14 ceramide and then we're like, oh, milk fat's really high in myristate, it was around that same time that SPTLC3 was discovered. So it kind of just naturally evolved together. And we thought, oh, well, we should look at the base too. So SPT's KMs for its substrates hover right around physiological conditions. So I think that increasing it can really drive production based on just the KM effect.
Okay. And since ceramide S5 and ceramide S6 have similar substrate preference, it's striking that only the knockout of her ceramide S5 didn't experience redundancy effect. So why didn't the ceramide S6 then didn't kick in?
This is a mystery to me. Ceramide synthase 6 has been manipulated in hepatocytes and it has really broad metabolic effects. Ceramide synthase 5 doesn't. So I think that when we're doing in vitro assays looking at their activities, there may be a species that one makes that the other doesn't that we've never looked at. What if a branched chain fatty acyl CoA could be on the N acyl chain? I think there are differences between the two that no one understands, but there's a fair amount of biological data that the two are very functionally different, despite them being able to use 2 or 3 of the same substrates in an in vitro assay.
All right, so these are all the questions that people have posted. Are there people who have additional questions that they would like to, post or comments? Could be comments. Verbally, then just raise your hand.
These are all such great questions. It's such a fun discussion. I'm
No,
really
it's a great, you see, you inspired us, and I really, obviously you are one of the forefront experts, so that was also an opportunity for us, really, and we are very grateful. Again, is there anyone else who would like to ask a question to our speaker?
I guess I have one more question. I'm
Okay.
always good for one more question. Milk fat has a lot of laurate in it too. Have you looked for C12s?
Yeah, we do. It's not part of our standard method, but yeah, there were differences in C12. It may be that the core we did those measurements in the I'm looking at the presentation because I can't remember if that figure went down to C12. It probably didn't. We were at Medical University of South Carolina at the time, and I didn't run that core. So we just used their standard method, and I doubt they went down to C12. And some cores use a C12 as an internal standard. That is one thing that we've learned is to maybe shift more towards deuterated standards, even though they're more expensive. A lot of the
Yeah.
original targeted lipidomics methodology was worked out using D17 sphingosine-based sphingolipids.
Yep.
Yeah.
Okay.
And so in some of my when I first started here about 20 something years ago, I inherited methods using C17 triglycerides as
Mhm.
internal standards in adipose tissue. And it turns out there's actually quite a lot of
Yeah.
tri 17,
Yeah.
a lot less 19, but it's there too.
Yeah.
So odd chain fatty acids are definitely around. They come in from ruminants too. foods.
Yeah. Yeah, because it's coming from their gut microbiota, right? Just like
Yeah, from the phytate
Okay.
decomposition. Yeah, so some of the plant fermentation yields 17 carbon metabolites in 15s.
Yeah. Have you ever tried to separate out whether it's a 16 methyl or a 17?
I know at least on the fatty acid side, once we chop off the chains, we can see that they're just straight saturates, not branched.
Okay.
At least the dominant form in adipose tissue
Okay.
is the saturate, not the branched.
Okay. We got really, really excited because one of our collaborators was working on branched chain amino acids in
Mhm.
heart, and we thought, oh wow, because they can be catabolized to substrates that will end up making branched chain fatty acids. And we thought, wow, that's occurring through, metabolic crosstalk through SPTLC3. And we went around and around. We developed the method to separate the two.
Mhm.
And in the end, like we just couldn't, we kind of dropped it because we just didn't see
Good.
much branched in the biology. The biology wasn't
Uh-huh.
branched was important in that way.
Good to know. I won't chase that.
Would have been cool though. And if you ever find
Absolutely.
something different, let me know because I'd love to have an excuse to pick it back up because it was.
So in a comment from my own side, and maybe in own interest, so we are hosting the MANA conference this year
Yeah.
Oh, wonderful.
at UC Davis.
Mhm.
So hope that you can attend. But for everybody on the call, the call is open for suggesting workshops and interactive forums. Where discussions are the main focus rather than presentations. So since we have here a really interesting discussion, maybe that would be also a topic to consider. And everybody, if you think that usually the conferences, they don't discuss the things that I want to discuss, that is your opportunity. We have the website open at MANA events, some Metabolomics Association North America. You go there. Until May 11, we invite people for ideas to add workshops and interactive forums. Could be on function of lipids, could be on function of single lipids, could be on function of our chain fatty acids, could be on, so these things are you need an audience, you need people who measure things, you need people who are interested in things. And there's, while you might sometimes have these very specific conferences, this is a slightly different way or a slightly different audience. Just think about it. Okay, about 300 people will show up.
Nice.
Okay, so are there further questions right now?
Just a fabulous piece of work. I mean, all of it was really, really, really wonderful.
Yes.
Thank you so much. It's great to talk about people who you don't have to argue about whether they're chain fatty acids with. So thank you.
All right. Okay, then everybody, thank you so much for attending, our April seminar. Next month, we will go back into data science and metabolism. We will talk about novel compounds and, everywhere where they come up, using AI tools for, elucidating the dark metabolism. That's coming up with this, Mike Skinner from Princeton University, end of May next month. And again, invites will be sent out. You can already register if you like. Okay, so thanks everybody for joining, and thanks again to Professor Cowart for an excellent talk, excellent research, and great discussion. Thank you so much, everybody.
Thank you. I really
Take
appreciate
care.
it. Bye-bye.